Okay, right, so welcome back to this next video in which we are discussing the local anaesthetics. Okay, right, so we've now seen the alpha subunit of a voltage-gated sodium channel. We've seen that the whole polypeptide has a membrane-spanning topology like so, and that these four domains that you have within this structure wrap around to form the 4/4 of the alpha subunit of the voltage-gated sodium channel, okay, to form the pore-forming unit of the voltage-gated sodium channel. Okay, and some very important structures are these P-loops and these S4 membrane-spanning alpha helices. Okay, right, we'll talk more about the P-loop and the S4 membrane-spanning alpha helix when we come on to the opening mechanism of the voltage-gated sodium channels. But before we get on to that, I want to finish our discussion of the structure of the voltage-gated sodium channels. So, the first thing I need to tell you is that there is not just one alpha subunit of a voltage-gated sodium channel. What would be ridiculously and beautifully simple is if there was just one protein which was the alpha subunit of a voltage-gated sodium channel, and that was the only one there was. But of course, in the human body, there is not just one alpha subunit for a voltage-gated sodium channel. Instead, there are 9 different types of alpha subunits of voltage-gated sodium channels. Okay, so there are 9 alpha subunits. Okay, and these have a very nice naming system. Okay, they are called NAV 1.1 all the way through to NAV 1.9. So the NAV portion stands for voltage-gated sodium channel. Okay, and then they've all got this 1, and then it's 0.1. Okay, and the point, the number after the decimal point tells you which alpha subunit you are dealing with. Okay, so we have NAV 1.1, NAV 1.2, NAV 1.3, NAV 1.4, all the way up to NAV 1.9 to give us overall 9 different types of alpha subunits. Now, all of these alpha subunits are coded by separate genes in the human genome. Okay, and those genes will all have different sequences of organic bases and therefore will code for proteins with different sequences of amino acids. So all of these 9 different types of alpha subunits will have a different primary sequence of amino acids. However, they are all similar enough that they all share the same membrane spanning topology. So they'll all span the membrane just like so. Okay, and they can all fold up to form the pore of a voltage-gated sodium channel, and indeed they are a functional voltage-gated sodium channel on their own. They will respond to depolarization, they will open and allow a current of sodium to move through them, and they will inactivate. Okay, right, so let me now discuss with you the tissue distribution of these different types of voltage-gated sodium channel alpha subunits, okay, because different tissues express different voltage-gated sodium channels, basically. So, which are the excitable tissues then? So, there is neuronal tissue, and then there is also muscle tissue. Those are the main 2 excitable forms of tissue. And by excitable forms of tissue, what I mean is a tissue that is capable, well, a cell type that is capable of conducting action potentials. Okay, so we have neuronal cells, and we also have muscle cells, and within muscle cells you have skeletal muscle cells and cardiac muscle cells, which are capable of conducting action potentials. Okay, right, so let's start off with neurons. So basically, you find NAV 1.1, NAV 1.2, NAV 1.3, and also NAV 1.6 are the main alpha subunits of voltage-gated sodium channels which you find within central nervous system neurons. Okay, so in the brain and spinal cord, the neurons will generally have voltage-gated sodium channel alpha subunits, which are either NAV 1.1, NAV 1.2, NAV 1.3, or NAV 1.4. Meanwhile, in the peripheral nervous system, so all neurons that are outside of the spinal cord, okay, so they have their cell bodies outside of the spinal cord, these these have voltage-gated sodium channels that have alpha subunits, which are either NAV 1.7, NAV 1.8, or NAV 1.9. Now, of course, this is an overgeneralization, okay, but it's a useful generalization. Okay, so these are the ones which you find within peripheral nervous system neurons. Okay, right. These then are the types of alpha subunits of voltage-gated sodium channels which you find within neuronal cells. Okay, so these would be called the neuronal types of alpha subunits of voltage-gated sodium channels. Okay, so these are the neuronal types. So all 7 of these, NAV 1.1, NAV 1.2, NAV 1.3, NAV 1.6, NAV 1.7, NAV 1.8, and NAV 1.9, those are all neuronal types. They're mainly found within neuronal tissue. Okay, meanwhile, the other 2 are then found in muscle tissue. So NAV 1.4 is then generally found within skeletal muscle. Okay, and that leaves the final one, which is NAV 1.5, which is then found in cardiac muscle. Okay, so these are the skeletal muscle selective ones, and then the cardiac selective ones. Okay, right. So those are all alpha subunits of voltage-gated sodium channels. They will all produce proteins of the same membrane-spanning topology, which will fold up into a pore-forming unit which can act as a voltage-gated sodium channel on its own. So, let's now turn our attention to the beta subunits, these auxiliary subunits that can be added on to the alpha subunits, okay, to moderate their function, and also localize them in different portions of the cell. Okay, right. So let's start off with the membrane-spanning topology of beta subunits of voltage-gated sodium channels. So they have a very simple membrane-spanning topology, especially when you compare them to the alpha subunits. Okay, so the amino terminus is extracellularly, like so, and then the carboxylic acid terminus is intracellularly. So they just have a single membrane-spanning alpha helix which crosses the membrane. And they then have an extracellular domain here, and then an intracellular domain here. So this is the membrane-spanning topology of a beta subunit. Okay, now there are 4 known beta subunits for voltage-gated sodium channels, and these are abbreviated NAV beta, so voltage-gated sodium channel beta subunit, and then you have the number, so you have beta 1, then you have the NAV beta 2, the NAV beta 3, all the way up to the NAV beta 4, which is the voltage-gated sodium channel beta 4 subunit. Okay, so those are the 4 beta subunits that can join onto alpha subunits of voltage-gated sodium channels. So let me show this. Okay, so let's go back to this picture where we draw those sort of more macroscopic structure. So here is our alpha subunit here, and we know of course that it's made up of these 4 quarters, which are the 4 domains of the voltage-gated sodium channel alpha subunit, all folded round to form this ion channel. Okay, and basically this is the rules for how you can join beta subunits onto alpha subunits of voltage-gated sodium channels. Okay, right, so off domain 1 over here, you can attach a beta subunit, okay, and this beta subunit will attach covalently to domain 1. Okay, so this is going to be here a covalent interaction. Okay, and the beta subunits which you can attach onto domain 1 of the alpha subunit of the voltage-gated sodium channel, it's either beta 2 or beta 4. Okay, so basically when you are building a voltage-gated sodium channel, the first thing you have to decide is which alpha subunit you're going to use. And then once you've got your alpha subunit, you then have to decide, do you want to include a beta subunit covalently bound to domain 1? And if you decide yes, you then have to decide between beta 2 or beta 4. Okay, so pick one of those and then stick it on. Okay, but you don't have to include one. You could decide, actually, I don't want one there, and don't attach it, therefore, onto domain 1. Okay, similarly, there is another site where you can attach beta subunits, which is domain 4. Okay, so another one can in principle attach to domain 4, and this is a non-covalent interaction here. So these two attach non-covalently. Okay, right, and in this site you can either attach the beta 1 subunit or you can attach the beta 3 subunit. So you've got your alpha subunit of your voltage-gated sodium channel. You've just had to decide whether you wanted to include a beta subunit bound to domain 1 of that voltage-gated sodium channel alpha subunit. Okay, and you've then had to choose between the two available. And now you have to decide, do you want to have a beta subunit attached onto domain 4 of your voltage-gated sodium channel? If you decide yes, you then have the agonizing choice of whether you want NAV beta 1 or NAV beta 3. So that's the structure then of a complete voltage-gated sodium channel with these beta subunits. And these beta subunits, by the way, are often called the auxiliary subunits, okay, or the accessory subunits. So they're not essential for the function of the alpha subunit. They just moderate its function slightly. And then, as I say, their main role is deciding where the alpha subunits actually end up within the cell, i.e. which portion of membrane they actually end up within. Okay, right. So, what we're now going to talk about is the opening mechanism then of voltage-gated sodium channels. Okay, so we've finished the structure then of voltage-gated sodium channels. What we now want to turn our attention to is how do they open in response to a depolarization in the electrical potential difference across the cell membrane. So let's go back to this picture where we are looking at the alpha subunit of the voltage-gated sodium channel from above. And I should just say, we don't care about the beta subunits anymore. The beta subunits aren't important with regards to this, or at least their importance is very poorly understood, so we're not going to discuss it. We're just going to look at the alpha subunit. Okay, right, so here is the alpha subunit. Okay, split into these 4 separate domains, like so. And then, as I said earlier, if we're looking from the extracellular aspect, the innermost portion of each of these quarters is going to be made up of the p-loop from each of those domains, basically. Okay, and these p-loops will line the pore at the extracellular aspect. And then, a little bit more peripherally, we then have the S4 membrane-spanning alpha helix, which is positively charged. Okay, and this is going to be very important in the opening of the voltage-gated sodium channel. Okay, so what I now want to do is draw a picture where I effectively take a cross-section like this. Okay, and I'm doing it deliberately like that so that I'm going to get the S4 membrane-spanning alpha helices within my cross-section. Okay, so imagine cutting down through the alpha subunit of the voltage-gated sodium channel as though it's a cake, basically, and then, of course, you're going to split it in two, and now we can look at the side, basically. We're going to look at the inners of the voltage-gated sodium channel alpha subunit, just as you'd be able to look at the inside of a cake if you cut it in half like that. Okay, right, so let me draw that picture now. I'm going to draw it sitting in the membrane. Okay, so we're now looking from the side at this cross-section. Okay, right, so first mistake, I've made it a little bit too big with regards to the membrane, but never mind. Okay, so, like so. Okay, so this portion up here, right at the top, this is made up of the P-loop here. Okay, so this is the P-loop in red here. Okay, then further out we've got the S4 membrane spanning alpha helix. Now, as I said, I've made a little bit of a mistake with regards to the size of the membrane. Imagine making the membrane bigger now, like so. Okay, so get rid of this. Okay, so that the S4 membrane spanning alpha helix is actually spanning the membrane. Then you've got the pore in between the P-loop there, and then this stretches in down here to actually close the channel, because at the moment we're starting before we've actually depolarized the membrane. So at the moment, the electrical potential difference across our cell membrane, let's say, is negative 65 millivolts, meaning that the intracellular compartment's electrical potential is 65 millivolts lower than the extracellular compartment's electrical potential. And again, this portion of the voltage-gated sodium channel alpha subunit is made up of that P-loop. Okay, and I think I'll change that colouring in just to a full colour in. Okay, right, and again we're going to see another one of these S4 membrane spanning alpha helices here. And the important point about these S4 membrane spanning alpha helices is that they are positively charged. They have positively charged amino acid residues every 3 residues. Okay, so in blue here, this is the S4 membrane spanning alpha helix. Okay, right. So, hopefully what you can appreciate is that the P-loop, which extends into the pore like this, it doesn't go all the way down, and that fits, because remember the P-loop doesn't go all the way through the membrane. It goes into the membrane a little bit and then comes back out. So this sort of narrowing that you have up here, this is only at the top portion. It's only at the extracellular surface, and this narrow little portion of the pore here is what's known as the selectivity filter. Okay, so the P-loop is responsible for the selectivity filter. Okay, so what do I mean by this? Well, basically, when this voltage-gated sodium channel alpha subunit actually opens, which it isn't at the moment, this is the closed channel, okay? When it opens, you're going to get a conformational change occurring down here, and these portions are going to move backwards, okay? So imagine these portions have now gone back to where I'm drawing this dashed line. So you're going to get a nice open pore here, and then when it's actually open, the narrowest portion will be this portion here, because the P-loops won't change in conformation when you depolarize the electrical potential difference. So they will still remain this narrow, okay? So this is the narrowest portion which the sodium ions have to move through, and this is the portion which makes sure that it is sodium ions moving through, not potassium ions or calcium ions or other ions. And it produces a channel which is around 10 times more selective to sodium than it is to potassium. Okay, so basically the channel is 10 times better at conducting sodium ions than it is at conducting potassium ions. So it's not perfect. It doesn't, stop a single potassium ion from moving through, but it is at least selective for the sodium ions. Okay, right. So let's now talk about how we are going to activate this alpha subunit of the voltage-gated sodium channel by depolarizing the electrical potential difference across the cell membrane. So let's say we do depolarize the electrical potential difference across the cell membrane up to, let's say, negative 40 millivolts. Okay, so now the electrical potential intracellularly is only negative 40 millivolts lower than the electrical potential extracellularly. Now, what does that mean? Well, when the electrical potential difference across the cell membrane was negative 65 millivolts, the resting electrical potential difference to have across the cell membrane, that meant that the intracellular compartment's electrical potential was 65 millivolts lower than the extracellular compartments. These positively charged ions in the S4 membrane-spanning alpha helix here, they will feel an attraction to the area of lower electrical potential, so they will be being pulled down towards the intracellular aspect continuously by the negative electrical potential difference that you have across this membrane. Okay, so you can imagine it's kind of like you're sort of pulling this down. Imagine it's an elastic band and you're stretching it down, and it's continuously poised in this sort of stretched manner, okay, and it's being held in that sort of uncomfortable position because you are continually having the electrical potential difference across the cell membrane at negative 65 millivolts. So these S4 membrane spanning alpha helices are being held, pulled down in an unnatural position, okay? They want to recoil back up, but they're being held there by the electrical potential difference across the cell membrane. When we suddenly reduce the negativity of the electrical potential difference across the cell membrane, what's going to happen? Well, it's going to be like releasing the elastic band, basically. Now that force of attraction that those S4 membrane-spanning alpha helices are going to feel for the intracellular compartment over the extracellular compartment is going to hugely reduce, and they're going to recoil back upwards, basically. And what's going to happen is when they recoil back upwards, they pull open this portion that is on the intracellular aspect and is occluding the channel. Okay, so what's going to happen is the S4 membrane-spanning alpha helices will move up, and they will cause a sort of pulling of this stuff inwards so that you open up that intracellular aspect of the channel, and you're therefore going to go from having this closed state channel to having an open state channel. Okay, so let me now show you this. So as I say, the P-loop remains completely unperturbed. This will still narrow this extracellular aspect, but this wasn't narrow enough to not allow sodium ions through. It was this portion that was blocking sodium ions from passaging through this channel, but now we're removing that portion. Okay, so it's changed conformation to something that looks more like this. Okay, so here now are these P-loops, and as I said earlier, you can now see that they they line the narrowest portion of the pore now, and they are going to be responsible for this 10 times selectivity for sodium over potassium. Okay, and now I'll try and show this. The S4 membrane-spanning alpha helices have moved up somewhat, like so. Okay, and this is responsible for the conformational change that has opened up the intracellular aspect of the pore, basically. Okay, so here are the S4 membrane-spanning alpha helices in blue here. Okay, and then the membrane continues on over here. Okay, so we now have the open channel. So this is the channel which will allow sodium ions to move into the cell within the up phase of the action potential. Okay, so that's explained how depolarization of the electrical potential difference across the cell membrane actually causes the voltage-gated sodium channel alpha subunit to open. But there is this more complicated thing that we now need to talk about, which is the fact that even though the electrical potential difference is still well beyond negative 40 millivolts, it closes, okay, after 1 millisecond generally. It only remains in this state for 1 millisecond, okay, and then it moves into another state known as the inactivated state. Okay, this doesn't fit with what I've told you, because so far what I've told you is that the reason the channel goes from being in the closed state to being in the open state is that these S4 membrane-spanning alpha helices move upwards when you depolarize the electrical potential difference, and that this opens up the intracellular aspect of the alpha subunit of the voltage-gated sodium channel to open the pore so that the sodium ions can actually move through it. But remember, the voltage-gated sodium channels are going to close up here. Okay, so at plus 40 millivolts, surely at plus 40 millivolts, you know, the S4 membrane-spanning alpha helices will move up even further, and that should open it even wider. So basically, you do not go back from this state into this state. Okay, they are not going from this state into this state at plus 40 millivolts. Instead, they go into a different state known as the inactivated state. Okay, right. So we now want to discuss what happens in order to take them from the open state, inactivated state, okay, from the open state to the inactivated state. Well, basically, this picture that I've drawn for you here is incomplete, okay? We need an extra structure to be shown here, okay? And this structure is known as the inactivation gate, okay? So there is a loop on the intracellular aspect of the alpha subunit of the voltage-gated sodium channel, which is going to be called the inactivation gate, okay? Now I want to show you where this loop actually is within the membrane-spanning topology picture of the voltage-gated sodium channel alpha subunit. So basically, this loop corresponds to the loop between domain 3 and domain 4 here. So this loop dangles down into the cytoplasm, and this is going to function as the inactivation gate. So let me show you how. So here it is in purple. And of course, it will have been in this closed state channel as well. And it will have been dangling down into the cytoplasm, perfectly happy. Now, basically, once the channel goes into the open state, what can happen is this loop between domain 3 and domain 4 of the voltage-gated sodium channel alpha subunit can end up going into the pore here and binding within this pore and blocking the pore, basically. So blocking sodium channels from, sodium ions rather, from moving through the pore. So let me show this. So here's the membrane, like so. Then we've got our voltage-gated sodium channel sitting in the membrane, okay, like so. And here's the P-loop here, making up the selectivity filter on the extracellular aspect of the channel, okay. And now we've got the P-loop the inactivation gate sticking into the pore. It binds within the pore on the intracellular aspect of the pore and is going to occlude the pore to stop sodium ions from moving through there. And this is how the voltage-gated sodium channel inactivates, basically. Okay, so these are the P-loops here in red. Okay, and that purple loop there, this is the inactivation gate again. And now we can see that this channel is not going to conduct sodium ions, and this is what's happening to the channels when they close, when the action potential is around plus 40 millivolts. But I'll stress again that they do not close because the action potential gets to plus 40 millivolts. This just happens, basically. As soon as you take the channel to the open state, what can happen is the inactivation gate can bind within the intracellular aspect of the pore and then shuts it down, and that generally happens in about 1 millisecond. Okay, right. So what I now want to discuss is the amino acids within the loop between domain 3 and domain 4, which are responsible for the binding of this inactivation gate within the intracellular aspect of the pore. Okay, so basically, if I get another piece of paper and draw this picture out for you. Okay, so let's revise our membrane-spanning topology of the alpha subunit of the voltage-gated sodium channel. Okay, so here is the cell membrane again, okay, with the two layers of phospholipids. Here is the amino terminus on the cytoplasmic side, so we're just drawing the alpha subunit again so that I can show you a sequence of 4 amino acids which are in this loop between domain 3 and domain 4, which are very much so important in the binding to the intracellular aspect of the pore of the voltage-gated sodium channel alpha subunit. Okay, so here is domain 1, then we have domain 2, like so, with its P-loop and 6th membrane-spanning alpha helix. Then we have domain 3, like so, S5, the P-loop, S6. Then this special loop between domain 3 and domain 4, which is going to contain a very special sequence of amino acids that I'm going to draw in here in a moment. Okay, and then we'll just complete it first. So we've then got domain 6, like so, with the carboxylic acid terminus intracellularly. Okay, like so. So this is the alpha subunit of the voltage-gated sodium channel. So I'll just put that here. This is the alpha subunit. So basically, within this loop between domain 3 and domain 4, you have a sequence of 4 amino acids, which is the IFMT sequence, okay? And this sequence is extremely important for binding to the intracellular aspect of that pore. Okay, so in order to be able to understand what IFMT actually means, you need to be able to read single letter amino acid code. Okay, so I, the first letter here, means that the first amino acid going from amino side to carboxylic acid side is isoleucine. So let me show you the structure of this. And I think it would be quite nice to draw out the actual atom by atom structure of this little sequence of 4 amino acids. Okay, so let's start off with the isoleucine residue. So here is the amino terminus of the isoleucine residue. Then we have the alpha carbon of the isoleucine residue. Okay, so at the moment I'm just drawing the core structure of an amino acid residue, and then here we have the carboxylic acid group, which will be involved in the involved in the peptide link to the amino group of the next amino acid along. And just as I'm drawing this carboxylic acid group as though it's involved in a peptide link to the amino group of the next amino acid along, this amino group over here is drawn as though it's involved in a peptide link to the carboxylic acid group of the amino acid that was before it. Okay, now the R group then of isoleucine consists of two main carbons, which then have methyl groups coming off. So this first carbon has a single methyl group coming off, and the second carbon also has a single methyl group coming off. And then we'll saturate everything else up with hydrogens. Okay, so that's the structure of this first amino acid going from the amino side to the carboxylic acid side, and it's an isoleucine. Okay, so the next amino acid residue is F. Now, what does F stand for? Well, basically, F is for phenylalanine, and you might wonder, well, why have they called phenylalanine F? Why didn't they call it P? Well, P went to proline, basically. So when we came to phenylalanine, we had to come up with something else. So obviously, because it sounds as though it begins with an F, we then chose to call it F. So here is the alpha carbon of this phenylalanine residue. Then we have the carboxylic acid group here, and then the R group of the phenylalanine residue then. So basically, it's called phenylalanine for a reason. Alanine is just the amino acid when you just have a methyl group coming off the alpha carbon. Phenylalanine is then when off the methyl group that comes off the alpha carbon of alanine, you then have a benzene ring, or a phenyl group as it's called. So, like so. This is a benzene ring stuck off that carbon, and that's what's known as a phenyl group, and hence this is called phenylalanine. Okay, right. Then the next amino acid along after phenylalanine is then going to be M. So what's the single letter M stand for in amino acid code? Well, it stands for methionine. Okay, right. So here is the amino group again. Here's the alpha carbon, and then the carboxylic acid group. That's just the core amino acid structure. And now the R group of a methionine amino acid residue consists of an ethylene group. So let's start by drawing that. An ethylene group is just two methylene groups joined together. Okay, like so. Then off the ethylene group you have a sulfur atom, okay, and then a methyl group off that sulfur atom, like so. Okay, so that's a methionine amino acid, and then the final amino acid then, so T, and I'm not going to be able to fit it in there, shouldn't leave myself the space there, so I'll put it up here. So T, the single letter T, stands for threonine. Okay, now basically threonine has a very similar structure to serine, except that it has an extra methyl group coming off of that carbon that comes off the alpha carbon. So let's start off with the core structure as always, and again that will be linked then to the next amino acid along, which we're not interested in. And then the R group is a carbon with an alcohol group coming off, but instead of just having two hydrogens coming off this carbon then, which will give us serine, instead you have a methyl group coming off here. So here's a methyl group, and then here's a hydrogen. Okay, so that's a threonine residue. So isoleucine, phenylalanine, methionine, and then threonine. That's this sequence of 4 amino acids, and this is the sequence which is responsible for binding somewhere within the pore on the intracellular aspect of our alpha subunit of our voltage-gated sodium channel, and it will then block the pore. It will stop sodium ions from being able to move through that channel, and that's the channel in the inactivated state then. And now, the important thing is that the channel will remain in this inactivated state until the membrane electrical potential difference goes down to the normal level. Okay, so the inactivation gate will only cleave off once the electrical potential difference across the cell membrane has gone back to the resting level. Okay, so when you repolarize the membrane, okay, so upon repolarization, the inactivation gate will cleave away from the intracellular aspect of the pore of the voltage-gated sodium channel. This process is known as de-inactivation. Okay, so the process of the inactivation gate binding within the pore was called inactivation. So this process going into the inactivation state was called inactivation, and the process of it unbinding then is called de-inactivation. And actually, when you do de-inactivate, temporarily you will go back into the open channel. Okay, so you'll go into the open state for a transient moment, okay? But then, of course, because the electrical potential difference is now back at negative 65 millivolts, what will happen is then the S4 loops will be pulled back down and you will close. So you'll go back from the open state to the closed state. So, looking at this in terms of the graph of the action potential, when we repolarize down to here, what will happen is the channel will move from the inactivated state back into the open state transiently, okay, and then it will move back into the closed state, and now it's ready to be activated again. So it's ready for a depolarization in the electrical potential difference across the cell membrane to cause it to open again. Okay, right, so that then concludes the opening of the voltage-gated sodium channel. Okay, so we've now finished our study of the voltage-gated sodium channel in detail, and we can turn our attention to the local anesthetic drugs. Okay, so we'll have a break here, and then in the next video we'll begin discussing the local anesthetic drugs.